Today I will be talking about routine surveillance of multi-resistant organisms. So Alison gave a really good overview of a comprehensive infection prevention and control program, but today we're looking at multi-resistant organism surveillance and how, I guess, we've interacted with the Queensland Genomics Health Alliance and incorporated that into an outbreak situation. Okay, so as Brisbane mentioned, I'm one of the clinical nurse consultants in the Infection Monitoring and Prevention Service here at the Royal Brisbane and Women's Hospital, arguably Queensland's largest hospital. That's what the website says anyway. So at any given time, we've got approximately 1,000 beds, and we're a tertiary quaternary teaching hospital. Amongst our many and varied specialties, we have got cancer care, maternity, neonatal intensive care, and an extensive trauma and burns service. And today I'll be focusing a bit more on our burns service and a particular outbreak we had in that particular unit in the recent past. So just to give you an idea of the activity we do here, in 2018 we had 78,000 or nearly 79,000 emergency department visits. Cancer care visits were just under the 70,000 mark, nearly 27,000 surgical procedures, and 4,600 babies were delivered, and that's about 12.6 babies every day along those lines. Okay. So I'm just going to step back in time a little bit just to give you a bit of context about the actual outbreak I will be addressing. And the reason I call it a cast of crabs was because that's the collective noun for crabs. And I'm really glad I'm speaking to an audience where I don't have to explain that I'm talking about pubic lice. So thank you. Is there anyone here who thinks it's pubic lice? No? Good. Lovely. Very good. So we're going to cast our minds back to 2016 to talk about carbapenem-resistant Acinetobacter baumannii. And this is an outbreak that occurred in our intensive care unit and burns unit. CRAB is basically resistant to the carbapenem group of antibiotics. It limits life-saving treatment options, and it has a mortality rate of around 30% if you get it in the bloodstream. Obviously, like any other infection, it complicates and extends admissions. Transmission is on unclean hands and contaminated equipment, and it's an environmentally hardy organism that can exist on unclean surfaces for months to years. And in fact, this particular organism was described as particularly sticky when observed under the microscope. Don't know what that exactly looks like, but it was apparently very sticky, so it adheres to plastic surfaces and that sort of thing quite readily. I actually can identify the patient by their antibiogram. That's a bit sad, really, isn't it, when you can do that? This patient was quite unwell for an extended period of time, but you can see by the resistance pattern just how resistant this particular organism is. I think it was only susceptible to colistin and tigecycline? Yes, yeah, it was very, very resistant. So this is how the the Great Crab Outbreak of 2016 occurred within our burns and ICU unit. Now, we've got some lovely diagrams, bioinformatic diagrams now, but this is traditionally how we used to do it. And that's only half the outbreak. We call it a table run or a Dead Sea Scroll. So you can see by this that it's a timeline, and you can see where the clustering actually occurred in both of the units, and it's colour-coded, so we won't go into too much detail there. But this is the breakdown over where the true outbreak sort of really occurred. So we can see the active transmission between June, July and August. And so on the 1st of June, we called our first multidisciplinary team, and we put into place our usual infection control measures like increased surveillance of patients in the environment, enhanced cleaning strategies. We allocated one of us, Michelle or I, to the outbreak coordination, and all patients in intensive care unit were placed on precautionary contact precautions. So they were added measures. There were other numerous strategies that were implemented to monitor and prevent clinical harm and disrupt transmission, including enhanced cleaning of patient environment, patient and environmental surveillance, as I mentioned, which extended into the operating theatre because it was feared that because these were related to burns patients, that there was a reservoir within the theatres. We did increased hand hygiene auditing. We observed workflows and work practices within the intensive care unit. People were observed in isolation rooms on their mobile phones, that sort of thing. So they were taking in extra equipment that could potentially have been a vector and a mode of transmission. And of course, there are executive briefings, weekly sit reps, consumer engagement, and on and on and on. So we flash forward to May 2018. The 1st of June. Hello again. Sorry. In my previous email, I meant to flag a new highly resistant crab that popped up this week. I've spoken to the lab and believe it will be sent for whole genome sequencing. It will be interesting to see if it's related to the great crab outbreak of 2016 isolates. Ooh, let's hope not. I've CC'd Brian to keep a lookout. So what's going on? We thought we'd got over the 2016 crab outbreak. What are we going to do next, and what are some of the things we need to do? So, in your packs, there is an activity, worksheet number 1. If you could, yep.
Which is it at this point in time now? Because I think we're just going to open this one up to the room.
Oh, sorry. Not the worksheet, that's later. Sorry. Yeah, that's later. Okay, so just have a quick discussion amongst yourselves and work out some of the answers to these questions, and then we'll just take some answers from the floor. So, yep. The next one. Yeah. That's the big one. Yes, and that one's yeah. Okay, yeah. Okay. Yeah.
We didn't end up doing a worksheet for this.
Oh, right. Okay, that's fine. Yep, cool.
Because when we did it, there was nothing to.
No, that's all good. Yeah, great. Oh, okay, that's all right. Not complicated. There's a lot. Yeah. They keep thinking that. Yeah.
Pleasing that found out in Melbourne Genomics alone. This woman has just presented, and they think they're leading the way in the IG space. Using genomics tracking transition of supercomputers.
It is a little bit long, yeah.
I'm just going to text Kate and say to her,
Okay, 1 more minute and then we'll get you to come back in. I can do it action shot again. All right. Have you all come up with some answers? Still deep in discussion. Okay. What are we thinking? What's going on? It's only a single case. Are we worried?
Very calm in our tone.
Very calm. Everyone's calm and relaxed. Excellent. Yes.
A little bit excited.
Nothing.
Screening.
Nothing triggering. Nothing. Yeah. Do a bit of screening. Yeah.
Environmental hygiene.
Excellent.
And just keep a close watch as what's going on.
Excellent.
We reckon there are 2 broad possibilities. Either this represents the crab outbreak strain, or this patient has independently brought crab into the hospital, so we want to do a history, have they travelled recently, what their exposure history, have they had procedures at other hospitals in Queensland or around the country or overseas, what's their story, and where have they been in our hospital, have they been to ICU, burns patients, so understanding the context of the patient.
Excellent. Wonderful. Okay. So this is what happened next. On the 9th of July, we received our QGHA report for our Acinetobacter baumannii, and it turned out that indeed it was the ST100 strain that was the 2016 outbreak strain. So, dear Crispin and team, please find attached the latest QGHA report covering sequenced gram-negative resistant bacteria strains at the RBWH. There are a few notable things. There are 2 new crab cases from 4C burns, which are identical to each other, which is evidence of direct transmission. And more worryingly, they are the ST1050 and closely linked to the 2016-2017 outbreak. Clearly, this problem has not gone away, and we would be very interested to hear your thoughts on how this has reared its ugly head again. Are there previously colonised patients that have come back to the ward, or could there still be an environmental reservoir that we are unaware of? So, what we did next was a bit of a literature search. Dear Patrick, thanks very much for the report. Lots of really useful information as always. We're doing an epidemiological review of the 2 crab cases to see if they have previously been colonised or if this represents new transmission. We never found the environmental reservoir, but if these results show that we have found an environmental reservoir, we need to go looking at the plumbing and potentially replace some of the bath drains and the proximal plumbing that was done in the crab outbreak in Canberra. So, yes, we did a literature search, which also yielded this. And so, this is a picture from my last holiday study. No, just kidding. I wouldn't be seen dead in a lime green bikini, no. So what lurks beneath? Okay, so when you look, it can be a little bit scary. All right. So we're starting to look in the environment, and it's not our usual targets of high-touch surfaces, bathrooms, toilets, that sort of thing. What specific things do you think we might sample? How would you collect? Hang on, you're going to have another activity in a minute, so save those answers. What's the best method? The logistics, what to do, how would you store those specimens after collection? We're lucky here, we can just run them across to the lab. You know, we turn up with, you know, a fistful of swabs and the rel goes, oh. No, we don't, we usually bring a head, we're nice. Resources, how would you resource a situation like this? Where would you get advice? And finally, does this require a procedure or a plan or a guideline? How do we do this? Like, is it just ad hoc? You know, we kind of just fly by the seat of our pants sometimes. And then finally, and importantly, what are you going to do with the results? So you do have a sheet this time. It's worksheet, handout 1, sorry. Can't see this.
Or worksheet 1.
Yes. Yeah, worksheet. So if you can just start to answer some of those questions, that would be great. And then we'll come back together and convene and discuss our answers and see if we can come up with some collective ideas. It's a great brainstorming opportunity.
Yeah, worksheet 1. So answer together, worksheet number 1. The first question should be, what specifically should be sampled?
Oh, that's 2.
Oh, is it? Oh, sorry, no. Yeah, so it is worksheet 2. Our team's got it confused, sorry. It's worksheet number 2.
Number 2. It's all good. It's all good. All right, we might finish up the table discussion now. I'm getting the move along from from Dana. So maybe 2 more minutes.
Prioritising top priorities they want to and that part of it. Yeah, obviously, yeah. Choosing follow around prioritising your top priorities from your responsibilities.
What resources should we have allocated? And where should we focus on these but I would do it with the lab so that you can correlate it, so that you take the right information back. To make it take practice at half-time.
Someone to read that I would absolutely call in a microbial test and then anyone else pick that as their top priority.
All right. Thank you, everyone. So what we might do now... Hello, everyone ready? Great, thank you. What we might do now is just go table by table, and if you can just give us your top priority answer, like the one that you consider the most sort of pertinent. So we might start with Dana over on table over here.
The answer to question 1?
Just what you consider to be the most pertinent response here. Just pick your favourite response.
Sorry, I was late. So did you guys agree on a favourite? In terms of sampling, we said the sink and high-touch surfaces.
Yep. Okay. Anyone else agree? Yep. Cool.
That's really good. Did everyone else come up with high-touch surfaces as kind of the sampling priority?
What we did is we went down to the ward and observed a lot of behaviour, looked at what was happening down there, looked at how the nurses were doing things, looked at the pan rooms and spent a lot of time, Trish and Michelle spent a lot of time on the ward just learning what their business was and understanding where the risks might be. So I guess sort of, well, not, was it mostly Trish's? Yeah, but so I guess just looking at what's actually happening, practice as occurs rather than practice as imagined, and spending a lot of time looking at that, I guess.
Anyone who wasn't involved in the outbreak, have anything else to add
Give it
to
away.
Yeah.
So I think, yeah.
Maybe look at the equipment. So again, with the flow of the department, you'd look at the common use equipment perhaps that hasn't been used since that outbreak, that's been stored and brought back out for whatever reason.
Great. Down the back. Anyone? Janice.
We're talking about utilising resources that you have. So talking to the labs about, how to sample, how to get them to them, what type of sampling to take. Also, because drains and sinks came into the conversation, talking to people like plumbing to actually give you a bit of a better understanding of what's going on behind the wall.
Great.
not just what you're seeing.
So anyone from the lab have any input about what the best sampling method would be? What would you collect with? Like, what's swabs? Kind of.
Just a swab, wet swabs are like gel swabs are better than dry swabs.
Yep.
And just call the lab first to let them know, perhaps like you need to prioritise those specimens, like especially if something's happening on Friday afternoon, like you need to make sure that someone's going to
Yeah.
like a skeleton stuff.
Yeah, so this is really important information for the infection control practitioners to know as, before we actually embark upon, a big environmental surveillance.
I think the other important thing from our perspective is we need to understand when the results are going to be back, so that because that when we set up our meetings, when it's no use in coming back Friday afternoon as well, because then you're not there on the weekends, they don't know what to do, and so that's important to factor in.
Yep. Paul, your take? Oh, sorry. Yep. No. Here.
I also wanted to point out, like timing of the swab might be important if the surface is really clean. If we think likelihood of getting bacteria is less, so couple of seconds, 10 seconds or 20 seconds might improve the probability of getting bacteria, and also rolling over the swab, not just.
So the technique of actually,
So rolling
swabbing.
over and timing, and also contact agar might help, like touch agar rather than swabbing. So getting sample directly from the surface with the touch agar. And some of the agar might have also in the broth, we can use antiseptic neutralizer, because in hospital, after patient move, we use detergent to clean, and also in the surface, we use chlorine to clean the surface. So when we culture, so there is if we use swabs, then swab antiseptic neutralizer still remain. So when we culture, then the antiseptics might inhibit bacterial growth, so we might miss out some bacteria that we want to recover.
Yes, there's been occasions where we've swabbed for VRE and the swab has gone through the DivaCleanse or the sodium hypochlorite or whatever the solution is, and it's kind of a bit of a pointless exercise. So that's very useful information. Thank you, Nahid. Paul, sorry?
One thing we mentioned was the other patients, and I guess presumably this is a routine swab because
Yes.
on swab.
Yeah.
So time the repeated surveillance swabs based on the patient flow, so if a rapid turnover in the ward, you get shorter, and all the patients are there for weeks, maybe lots of weeks.
Yeah.
The other thing we mentioned in the context of this forum would be metagenomic sampling of surfaces.
Yep. Great. Lovely. Okay. Right. Thank you. All right. So before I talk about what happened next, I'm just going to give you a little bit of a tour of our burns unit. So I haven't got a pointer? No pointer? No, that's all right. Okay. So basically, here is the entrance to the ward, and there's 7 single rooms, 2 4-bed bays, and then another 3 single rooms at the top. And in the centre is where the burns baths are, which are a critical part of burns recovery. Yes? Oh, can I? Oh, I can. That's so good. There we go. All right. So entrance to the ward, nurses' station, single rooms, 4-bed bays, 3 single rooms, and then these are our 3 burns baths. And as I mentioned, these are critical to the patient's recovery from their burns injuries. So on the 12th of July, dear Patrick and team, the 2 new ST1050 crab cases in burns are new. It is likely that the first case was acquired the isolate within 3 days of being admitted to the ward after transfer to ICU to bed 1 in the burns unit, which is here. And the second patient then became colonised a few days whilst in bed 2, overlapping with the patient in bed 1. Now, from an infection control perspective, if you have 2 patients come up in the room next to each other, you would naturally assume hands or contaminated equipment. So we thought maybe that's how transmission was occurring. But But we did press on and we did do some environmental sampling, and we collected 2 sets of samples of each of these targets. So one was going for micro and one was going for molecular sampling, as Paul mentioned. Now, when we did do the sampling in the 3 bathrooms, so we looked at the shower head, the bath drain hole, the interior and the exterior surfaces. We took water samples from the bath waste. We looked at the floor trap at the underside of the grate. We took water samples from down at the water seal in the drain in the floor. And then we looked at room 1 and 2 where the 2 positive patients came up. So yes to all of those and responses that were just given. So that's great. So the patient environment, the hand wash basin, the toilet, surprisingly one of those came up positive. The ante-room to the environment, so the ante-chamber into the isolation room, as well as the sink in that room. Now, these are just some of the observations that we made whilst collecting these samples. So we noticed that there was, a fair bit of bioburden on the floor drains, which you'd expect, but particularly so in bathrooms 1 and 3. Patient support officer came up to us while we were swabbing. They were obviously naturally curious of what we're doing, and they chimed in and said that the floor traps in the patient bathrooms were also grimy. In those ante-rooms, it smelt a little bit musty and a little bit organic, so we thought, maybe there's something there. And they were cluttered with equipment, as you said, Desley. And when we went to collect the water sample from the floor trap, it's actually a really long way down to the water seal, and I didn't fancy putting my arm down, so we decided that we needed to get some good old-fashioned suction equipment. So that's what we did when we suctioned up and got lovely water samples from yeah. And here are some lovely pictures that we took on the day that we sampled. So this is the floor drain, as you can see. We had 2 plumbers with us who were very, very helpful, but also very, very worried that they were going to pick up crabs. So we had to explain, no, they were fine. The next one is particularly grimy. Down in the grate, you may be able to see that there are staples from surgery. There's obviously a lot of what looks like dirt, even mud, so it's a very moist environment. There's hair, there's probably skin and other bits of body substance. So yes, that was particularly interesting. The figure on your left is the burns bath itself. So this is what we bath our patients in, and the pinkish tinge is chlorhexidines, and that's what they bathe the patients in. The next picture is what the plumbers dismantled from underneath the bath. So burns patients are often lathered in paraffin. That's what they put on their burns, and so that's what you can actually see on the inside of that drain. And the pinkish tinge again is the chlorhexidine. And if you look really, really closely, there's other staples and the other bits and pieces that I mentioned just before. And here's an action shot of suctioning up from the floor. So this was interesting. I haven't suctioned in a really long time, and you can So it's about that far down to the water seal, so you can imagine that you don't want to stick your hand down that far. And that's what we did there. Oh, hello. Okay, so what did we find? Imps found an IMP4-like organism, but no crab. We didn't find crab. So burns bath 3, the floor trap, burns bath 1 under the grate on the internal side of the trap, burns bath 2, and so we found these organisms. Some grew the IMP4 and some didn't. And that was really interesting. So that were the micro results that we found there. Okay, so we move forward to August the 1st, and we get our 3rd case of crab. And this time it's a clinical sample. It's actually from an ear wound. So we have our first sort of infection as opposed to colonisation. And this patient came up in a 4-bed bay, which, was cause for concern because we were worried that there would have been further transmission within that 4-bed bay. They share the same bathroom, they share the same toilet, great places to have transmission occur. And then there's the floor trap. So on the 10th of August, we actually pieced together, I didn't, we pieced together the crab genome and from a floor water sample in the floor, and it did actually sequence the same type as the ST1050. Now, my explanation would be as simple as that puzzle piece there, but I'm going to call on Brian now to give further explanation about the metagenomic sequencing and bioinformatics analysis. So thank you, Brian.
Hi, everyone. So I'm Brian Ford. I'm actually a postdoc in the Scott Peterson's lab at UQ, and I do a lot of the bioinformatics for this. But I should say that for the crab isolate, apart from what I'm going to talk about now, most of the bioinformatics was done by a former student of ours, Leah Roberts, who's now with EBI in the UK. So before I start, I just want to just mention a couple of things. These might come up in the next couple of slides. So this is some of the techniques we use to find what Trish was looking for. So the first is it's called a minimum hashing or minim hash, and that's just a computational technique, and that quickly estimates how similar two different sets are. And it's used a lot in comparing websites. And I don't know for certain, but I wouldn't be surprised if it's used in stuff like Turnitin and stuff like that at UQ. The second one is a k-mer. So a k-mer is all possible substrings of length k that are in a string, and so pretty much a nucleotide sequence of a certain length, and we use this kind of method to look for identity. What I'll talk about a lot is a tool called Kraken, which is a taxonomic classification tool, which was mentioned a little bit earlier, and that uses k-mer matching. And then MAGs, which are metagenome assembled genomes, and these are like species-level genome assemblies. So they're not based on isolates, they're based on all members of a particular species that might be within a metagenome sample. So they're not really representative of an isolate, but more of the population level assembly. Caitlin earlier on, she spoke showed a bit about the pipeline that we normally use for isolate sequencing. So for this particular metagenomics, we did something a little bit differently, and we weren't very experienced in it. So what we're going to show you is really an ad hoc approach to what we did. It was really kind of trial and error because we weren't sure what we were going to get. So we're kind of really working on this right now to establish it and make it more robust. So first of all, what we did is we wanted because these were crab and we had a complete reference genome, we really wanted to see if we could use the raw sequencing data from the metagenome sample to see if our particular isolate of interest was there. So we had a complete genome, one of these. This MS14413 is a complete genome from the previous crab outbreaks that we sequenced. And we used a program called MASH, which uses the minimal hashing. And what we did is we used the MS14413 as a reference, and we screened all the reads from the metagenome sample against that one reference. And that gave us what is essentially a MASH score, and that gave us some level of an indication of whether our genome of interest was present within this larger metagenome sample. So we screened lots of different metagenomes from different sites, and we got a few promising hits. I don't have any fancy graphics to actually show you in this. It's just numbers, so I'm not going to put them up. But at the same time, we did another approach, so that was using the tool called Kraken, and this gives us community-level profiling of the metagenome samples. So what we did is we took all the raw reads from each metagenome sample, so these are just the raw sequencing data, and we screened them against the RefSeq database from NCBI, and that's a database, a really comprehensive database of bacterial genomes and viruses, I guess, and some fungi. And this is what we saw. So what we're seeing here is each column is a single metagenome, and so each different colored segment of that column has a percentage attached to it, and that percentage gives the abundance. So I guess it's pretty much the abundance of that particular species, which are color-coded, and you can see them down here on the side, what percentage that was of the total metagenome. So the percentage, though, is a percentage of the total number of reads that match to that particular genome. So what we could see in the vast majority, we could see no crab whatsoever. But in 3 of the samples, particularly in this one here, so these are highlighted with the black box surrounding them, we can see 3 have actual crab genomes within them. We can isolate Acinetobacter genomes, I should say, rather than crab. So this tool really gets us down to the species level. It can give us some indication of the strain level, but realistically, it gets us to the species level, basically. So we know there was Acinetobacters in these samples. From our MASH score, we had a good idea that these Acinetobacters were related to our crab outbreak strains. We didn't know how related they were because we were very inexperienced in MASH. So what we did is, from this program Kraken, we can extract all the reads that match to this particular genus of bacteria. So we take Kraken and we extract all the Acinetobacter baumannii reads from the metagenome data set, and then we mapped them to our reference genome again. And this gives you an example over here what this map would look like. So typically what we hope to see when we map across the genome is this whole top bit here would just be red, so we have good coverage across the entire genome. Every segment of the genome, which is represented down here, these are genes, and see genes here and proteins, so 6' translation. So everything in here is intergenic. So we would expect that in a normal genome sequencing, this whole region will be covered by reads. And what we can see, it's quite patchy, so we're not getting full coverage across the entire genome. But we are getting coverage across the genome. And what's represented here is some of the genes we'll see in MLST profiling. So when we extracted all these reads, so we have all the reads that map to our reference genome. So these are the only ones we're interested in right now. So these are the ones that we can say are Acinetobacter reads, but also should hopefully, if present, contain reads that are specific to this crab outbreak isolate. So then we tried to screen these for MLST, and it didn't actually work using the raw read. So we did the de novo assembly, and this is kind of this MAG assembly as I was talking about. And from this assembly then we could actually do some sequence typing. So we can use our different software to check for sequence type, and we got back what we thought was roughly the ST1050 allele allelic profile. So that was, for us, that was sufficient enough for us to say that this was most likely the crab outbreak strain. And then we also did some phylogeny. So we can't be certain from this because there's some errors in the allelic profiles that could indicate that it's a different sequence type and not related. But we did some phylogeny as well. So this is what the phylogenetic tree looks like. So up here in yellow is a bunch of unrelated crabs. So these are crabs we downloaded from the internet. And in here in pink, these are 2016 crab outbreak isolates. So what we were looking at for our isolates that we assembled from the metagenomes are actually down here in blue. And in green here, these are nanopore genomes that we assembled without doing any error correction, which is a completely different story. So what we can see, though, we can see that even that our crab isolates have a big, long branch that stretches out from the outbreak isolates, basically. But they do cluster within the outbreak isolates. So this is a clear indication that they are actually related to these isolates. The reason for the long branch, obviously, is because of all that missing DNA that we saw in the alignment. So we're missing a huge amount of DNA when we do a comparison. So there's obviously, and there'll be lots more kind of unrelated, but from reads from related species, which are so basically from the broader group which the crab belong to. I mean, would also be in here too. So we've got a lot more SNPs and stuff that we shouldn't normally see. But when we try to do some downsampling up here, we can see as well that when we do some downsampling of normal isolates, we do get extended branching moving out, not as obvious and evident as we saw in these ones, but it's just its placement within this group is clear indication that it's part of this outbreak. I think that's it for me.
Any questions for Brian before we move on to the rest? Sorry, Crispin has.
This is such an exciting month in the outbreak because we've done, I think, 300 standard culture-based swabs.
Okay, there's lots.
Hundreds and hundreds of standard culture-based swabs have haven't found Acinetobacter anywhere in the environment, despite looking very, very hard. We really strongly believe there was an environmental source. We just didn't know what it was. So this direct from swab to the main culture step was a really big step for us. It was a very exciting result, and really I think it's the fundamental reason why we can solve this outbreak. It's a big moment in our outbreak. So actually, there's a deeper history of the crab at Royal Brisbane. There have been the crab isolates at least since 1999, which is probably when we started finding them. So this has been going on for even longer than this time.
Yeah, no, that's great. So moving further into the outbreak, on the 13th of August we came across our fourth case of crab, but this time it was in a blood culture of a patient who had 80% total body surface area burns. So she was already a very, very, very susceptible patient, but it's our first, second, third, and then our fourth case back in bed too. So this was the next escalation point for us because this, if this patient died, that would have had some fairly significant ramifications. So I'm just going to open it up to the floor to say what happens next when you have something like this happen in your facility.
did the funders sort of suggest that particular areas drain to that floor trap, or does the entire unit's plumbing drain into that floor trap?
It was in the
Yeah.
bathrooms.
It was in the Burns bathroom, so
That was coming down.
yeah, what was coming through that lovely purple gunk was going down into that floor drain, yeah. Yes. Anyone? Crispin, go ahead.
I guess one thing now is what potentially you have is a patient who's undisputedly acquired, with no question, a hospital-acquired infection that they might cite from. So the whole concept of becoming a coronial investigation and medical legal is become very important now. This is one that we would hope. One of the things we've got to be very careful, and obviously it's a really good thing for the world and patients, but there's zero plausible deniability now. And 100% for sure say that this patient got this bloodstream infection from Royal Brisbane on this day, and our fault, and the medical, legal, moral and ethical implications of that are very, very important. It also really, really gets the executive interested in something like this in a way that, in terms of asking for funding and resources in a way that nothing else gets.
When they get a SAC one across their desk, they're all very interested. All of a sudden, they don't like SAC ones.
We've talked a lot about this, that this is clearly a SAC one. There's no possible implication that's not from us at this stage. If someone gets an MSSA bacteremia where we have a suspicion that it's from the hospital, that's a bit of a different situation. There's no wiggle room.
You have to stop movement in and out of the ward at this point.
Yes.
And ward can shut it and fix, obviously, a
Yeah.
Burns ward service in the state.
Yes. So what we did next was to brief up. So situation reports, SBAR, is everyone familiar with SBAR? Yeah, it's part of communicating for patient safety. It's a technique that we use to communicate succinctly and quickly a situation that is quite rapidly evolving. So S for situation is that we had active transmission of crab that was detected in 4C Burns, and since May the 28th, 2018, we had 5 patients identified with crab, but 3 in the previous week. Our assessment or action was to form the outbreak management team, and then I'll just go through our recommendations. So the recommendations we developed was to cease using Burns Bath 2 until further microbiological investigation was undertaken. Contact precautions for all patients in 4C and all patients transferring out of 4C on contact precautions, which means they would go back to another ward and be placed in isolation and require some clearance screens. So that would be 2 sets of rectal swabs 7 days apart, and if both of those came back negative, then they would be taken out of isolation. If they came back positive, they would go to 6C Wattlebrae, which is our infectious diseases ward, or if they required ICU, CCU or Burns, they would go to those units based on clinical need. Precautionary contact precautions were also kept in place for all transfers to theatre. So it shouldn't, but it does create a logistical nightmare in that area. Diagnostic testing as well as allied health appointments, so physio, OT, all of those things are really, really important and part of a patient's Burns recovery. And if they can't go to those things, it sets back their recovery quite significantly. So that was important that went continued. Additional cleaning support was arranged for the early shift, and that continued into the weekend also. We increased our hand hygiene auditing, as that's often a mode of transmission in any outbreak situation. And we did further environmental surveillance, which we undertook in Burns bathrooms 1, 2 and 3 again, and in patient bedroom number 2. And these results, so we collected again the 2 sets of swabs microbiologically and molecularly, and they were processed as well. We did try, just going back to what would you sample with, we did try 2 sets of swabs, so just your traditional blue cap swab, dipped in a bit of saline, and we also used these ones. So these are used quite often testing kitchens for their microbiological testing. If you are going into a patient room and it's somebody who gets a rectal swab weekly, it's really good to preface the conversation that this is not for you, because it's a little bit bigger. So, yeah, because it does draw, yes, some distress from patients.
Can I just say,
Yes.
all the time, but just with Paul's comment there, we did contemplate closing the ward. So during a crab outbreak in 2004, we were looking to close the Burns ward because the statewide service has no backup, and so we're all set to start sending our patients to Sydney. That has massive implications that I'm here to do that, but there isn't anywhere else really for them to be managed, but for severe events at least.
Yeah. So yes, as I mentioned, we did repeat our environmental surveillance, but we found no oxo, no imp. And on the 20th of August, we had our 6th case come up. On the 28th, the plumbers commenced their drain maintenance. So this is something that's routinely done in places where there's dialysis or chemotherapy, that sort of thing, just to clear the drains of those sorts of the stuff that builds up in the drains there. But we decided to use it as a mitigation strategy to make sure that there was, to eradicate the crab and the other organisms that had the imp4 in them down the drains, because obviously there was a clear connection. The way that they do the drains is quite interesting. That little blue tube that you can see coming out of the drain is actually attached to a rather large bladder, I guess, that they inflate with the bike pump. That goes down to the water seal, sits above the water seal, and then they pour down sodium hydroxide or Drano, and they leave it sit there for 30 minutes to take effect. They clean the drain covers and that sort of thing, and then they flush the drains after that. So that was quite an interesting exercise to see that in practice. On the 30th and the 31st, we had our 7th and 8th cases, both in wounds again, which is a concern. And so our risk mitigation strategies were sort of put under a bit of pressure when there was increased bed pressure on the 5th of September. As you can imagine, that's sort of coming to the end of flu season. We're always in code yellow. We're always over capacity. So we were forced to sort of pull back some of our infection control strategies, such as letting outliers come into the ward. We obviously risk mitigated that by saying nobody who's got is immunocompromised. What else did we say? Brisbane, open wounds, that sort of thing. So yes, it was just your garden variety medical patient sort of was the exception to the rule. Oh, Brian. So I'm going to call on Brian again to explain just the third lot of testing we did. Did you want a microphone, Brian?
Right? Yeah, no, okay.
Oh, good. Okay.
Don't know why I put this up. I think it's just to highlight that for some of the later isolates we got, even though they've still got big long branches, so this is thousands of SNPs. So ordinarily, if you saw this, if you were just to compare in terms of SNPs, you would say this is not nowhere related to this outbreak. But we put it in the context of a phylogeny, you can see that because it groups within this broader outbreak, we consider this related to these, and this long branches we're seeing are mainly as a result of us extracting the data from a metagenome rather than isolate sequencing itself. And then we have some other ones here as well that actually are these. We get a lot better assemblies out of these. I think we got a lot more reads in terms of the reads that they did. I'm not sure if they were. I'm not sure why. So maybe they were just more pure cultures. Yeah. Perhaps. I can't. Oh, actually, these were isolates that were extracted from the metagenomes after we identified them. So we can just see that they do actually group down within this group, and they're pretty closely related. I'll show you something else. Yeah, and this is just another way of visualizing that same outbreak. So what we have in here, so everything, the circles represent different isolates. Everything within a circle is identical at the core genome level. So we have large groups, so we have large groups of nearly identical isolates. The lines that connecting them means that they're related, but where it's not indicated, each line is one SNP. And then when we have more than one SNP, it's indicated by the number. So most of the isolates are related, they're one core genome SNP separated from each other, but we have some which are a little bit more distantly related, but still under 5 SNPs. So these ones here in the orange, these are from the 2016-2017 outbreak. And up here in the red box up the top, up here, in the yellow, these are the isolates we've got through the Queensland Genomics, the continued surveillance. We have about 10 isolates in total. What's interesting about these is that They do all cluster really closely related to each other, but they are separated by 3 SNPs, so they're distinct from the original outbreak. And then when we started doing the actual metagenome sampling, we can see that one of our metagenome, so one of the isolates we extracted or cultured from one of the metagenome samples, we can see it actually groups in here. So it's M8825, and this is one of the very first samples that we collected in the crab outbreak. So we can see that this is clear. I don't know if it can make it any clearer that this is definitely the source of, or at least one of the sources of the outbreak, this particular outbreak.
Thanks. Oh, yep. Okay. Moving into late September. After a pleasing few weeks with no crab isolates detected, another patient colonisation has emerged in an ICU patient. His exposure to the burns bath and unit in general is minimal. He's also been to theatre several times. We're going to get molecular typing done, but we are unlikely to get a result until next week. However, there is a very strong likelihood that this will be an outbreak strain. There's also another haematology-oncology patient in 5C, that's our hem-onc ward, with a crab isolate, but there's no epidemiological links to burns ICU or theatre. And this isolate has been sent for typing, but it's not likely to be related. The mere possibility that this could have been a related case with no apparent links was very, very, very concerning indeed. So having the reassurance that it wasn't linked was fantastic. So a very hurried phone call to Hawken to make sure that that particular isolate had got on the run out to FSS. Fortunately, Amy came to the rescue, and the sequence showed that it was a different ST type. So we could stand down in the haematology-oncology ward, and as you can imagine, that would have significant impact in that unit should we have had an extension of the crab outbreak. Okay. Finally, our closing meeting of our crab outbreak management team meeting was on the 22nd of October. So we set the parameter that if we had 4 consecutive weeks with no transmission, that we would declare the outbreak over, and fortunately we did at that point. It did have some flow-on and lasting effects, so we lifted all restrictions to admissions back to the ward. Hibiscus alerts were removed from patients who were associated with the outbreak, so we put on a precautionary contact precaution alert during these sorts of outbreaks, so we're able to remove that in patients. It wouldn't affect their patient placement then. Additional cleaning resources remained permanently now, and they still do remain permanently in 4C to support cleaning there. And I have to say that ward is meticulous all the time, and the hand hygiene is fantastic. Their compliance is always above 80%. Weekly drain cleaning continued in the baths until the plumbing was replaced. Routine surveillance of 4C, environmental surveillance, was to continue. And we were going to reconvene if we had 2 or more epidemiologically linked cases of crab, and fortunately we hadn't, so that's good. So this just maps out the crab outbreak between 2016 and 2018, and kind of presumed that we had 2 outbreaks. It's interesting to look at where the interventions occurred and where the outbreak actually ceased. So during the 2016-2017 intervention, the interventions commenced in about mid-June, but it took several months for that outbreak to actually sort of peter out. Whereas when we did the drain maintenance, the preventative maintenance in August, the outbreak was pretty well declared over quite quickly, so probably just over a month's period, in a month's time. So I guess it really begged the question, was it 1 or 2 outbreaks? Was it just burbling there over time? Was it the fact that we found the environmental source, that we haven't had any cases of crab attributed to ICU or burns since the 28th of, since the end of 2018? You know, I think environment certainly plays such a huge part within outbreaks, and particularly with this organism, can't be underestimated. I did have a bit of a dad joke, and I said crab is very draining, but anyway. Okay. So over the course of this particular part of the outbreak, which lasted 19 weeks, we did 3 rounds of environmental swabbing, which all had molecular as well as microbiological analysis. There were 8 cases and fortunately no deaths. I think we were very, very lucky, particularly with that case of the 80% burns. The impact of rapid identification and response to outbreaks with targeted infection prevention and control strategies can't be underestimated. If you look at service delivery and patient flow, burns units routinely take surgical and medical outliers. We've always got bed pressure. Decreased bed capacity within burns or, as Crispin mentioned, closure of the unit has the potential to significantly impact the hospital's overall capacity and patient flow processes. If transmission hadn't adequately been contained and spread had continued, we may have needed to have shut the unit, which, severely affects our ability to provide a quaternary service with a major catchment area. And if we'd had another major incident, such as, Ravenshoe or Barley, anything like that, our next closest burns unit is down at Royal North Shore. Environment and equipment. Environmental samples taken from the bath and the floor plumbing were found to be carrying the same plasmid found in the 26 crab outbreak, as Brian has described. We lost 30% of our burns bath capacity for that period of time. It took 4 months for those parts to arrive from overseas, and that was from New Zealand. Resourcing. I guess we did have a positive out of our outbreak. The additional cleaning has remained within 4C, so it really justified that. We wanted to establish an outbreak response cost centre. Sorry, I was having a little chuckle to myself because we're still waiting for that. The financial implications, I believe in one of the earlier presentations we talked about economic evaluation. That's really, really important. It's what did it actually cost versus what's it worth, to the organisation. Reputation. You risk the organisation's reputation if an outbreak can't be contained, if there's deaths associated, obviously. Patient risk, it impacts on operations and the ability to provide a burn service to a large catchment area. We also prepared a media holding statement, and that's become standard practice now with any outbreak that we have due to, the courier mail test, I guess. And at the centre of it all is always the patient. With a 30% risk of mortality if you get a bloodstream infection from this organism, with limited treatment options, extended length of stay, isolation and rescreening, from their many readmissions, burns patients come back time and time again for surgery. There's a significant personal cost to complex patient care. So routine surveillance, is it routine? Well, it's an evolving thing. The day in the life of an infection control nurse is not typical. It's never predictable, and it's rarely dull. We're entering a new era of infection detection. So whether it's routine, something oddly unexpected, or explosive, as we're about to find out in the next two sessions, the aim is always the same: preventing infection and avoiding patient harm.